Here we will cover how to upload your FTMS data to FREDA if you are not uploading data from CoreMS. If you are uploading data from CoreMS, skip this video and head to CoreMS Upload. When opening the FREDA application, you will be greeted by this screen. Click Single Aligned File and then go to the Upload tab. As covered in the Data Requirements video, there are two files. The first is the data file, which we'll upload by clicking this button here. We'll be using the same example data that was downloaded in the previous video. After upload is complete, FREDA will prompt the user to select the column that corresponds to the required mass identifiers column. In this case, it's mass. Now we upload the second file, the molecular identification file. And as you can see, a table summary appears here and shows that it has the same unique mass identifier column. Now we're prompted to select whether a molecular identification file has formulas or elemental columns. In this case, we have elemental counts for carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. We select elemental counts and are then prompted to select which columns identify those counts. Carbon and hydrogen are automatically detected, and the rest can be added with the input widgets here. You'll be asked whether or not your molecular identification file has information on isotopic peaks. In this case, we select yes, and that we do want to filter these peaks. You're then prompted to select which column contains this information. In our case, it's the column C13, and a 1 indicates the presence of this data. Once you've filled out all this information, you're ready to create the data object that will be used in FREDA. Click Process Data. The object is now created, and you're prompted to either review this page or continue to subsequent tabs. There are summaries on the numbers of peaks in your data, the number of samples, and the number of peaks with identified formulas. Your data is now successfully uploaded. In future videos, we'll go over how to perform further preprocessing steps such as grouping and adding metadata.